All right, guys, welcome back to episode 4 of the Harry Matters podcast. I'm Averbs123, world's greatest moderator, here with Andy1, who's a little bit shaken up after his girlfriend took him out for the first time in a couple months for the 4th of July. He's back at it because he wants to keep her around, and he needs that full head of hair to do it. Uh, we have a really awesome episode for you guys. First, we're going to jump into all of the community's favorite gene targets. A ton of them are brought to you by the world's greatest plumber scientist, Jumpman. And then actually big shout out to Benjels as well, who's gotten in touch with the world's preeminent researchers on many of these targets, including the mother of Twist1 and also one of the really top PII1 scientists. Then we'll get into some Paus fuel. Well, Ralf is joining us from Italy. He's going to talk about his next proposed study, which is studying thyrotropin-releasing hormone, TRH, which pairs really nicely with the first study that we conducted, T3, T4 thyroid hormones. He does take a shot at the Labdao community, may or may not have called us brain dead for our comparative dutasteride delivery vehicle study. I'm going to make the case later on why I think it's actually a good idea. And then we'll wrap it up with Nicholas, who is the co-founder of Labdao, and he's built the Plex software that we are using to design new drugs to target the targets that we're going to talk about at the beginning of this podcast. So with that, I'll give it off to Andy1, and he'll take us away with the first target.
At the top of the list, Twist1 remains our top target. The protein is a transcription factor, which means that it binds to specific regions of our DNA to control the activity of other genes. Transcription factors are notoriously hard to target, but obviously the challenge is our opportunity. Getting to the mechanism of action, Twist1's upregulation causes the downregulation of proteins that maintain cell-to-cell adhesion and also cellular identity. So again, keeping cells together and in the state that they were designed to be in. In our hair follicles, Twist1's upregulation causes our dermal papilla, which are thought to be the most important, if not the second most important cell group in our hair follicle, to lose their identity and migrate from their niche. And as our dermal papilla niche shrinks, so do our hair follicles. Hence our belief that Twist1's downregulation may result in hair growth. Besides hair loss, high levels of Twist1 are also associated with various forms of cancer. And so knocking down the gene may actually provide additional health benefits, which is in stark contrast to most potential hair loss treatments, which, as we were talking about before, tend to carry with them risks of cancer. There are downsides to regulating Twist1, the biggest one being that it may slow wound healing. So if you're having a surgery or in some sort of accident, God forbid, you probably don't want Twist1 downregulated too much in that scenario. So our goal there is thus to devise a treatment that has a very short half-life, meaning that after you apply it, even ideally we can apply it topically and get it to the cells that we want to get it to, the dermal papilla, and potentially the dermal sheath, which is the neighboring cell group to the dermal papilla. But then after it hits those cells, ideally it hits the blood and disintegrates, or degrades rather. And so we don't feel any systemic effects. Currently our community is in talks with several CROs to conduct screenings of ASOs, siRNAs, and even small molecules that we can apply both topically and orally to downregulate Twist1.
Twist2, while we feel really confident that we want to inhibit Twist1, it's really a flip of the coin what we want to do with Twist2, upregulate, downregulate. We're not really sure. On the case for upregulation, Twist2 has been found more highly expressed in hairy scalps versus bald scalps. Additionally, its knockdown upregulates our prolactin receptor activity and TCF4 and 12 proteins, both of which are bad for our dermal papilla. On the case for downregulation, Twist2 is oncogenetic as well, meaning that it could cause cancer again, which leaves us in a tricky position if we're trying to upregulate it. And then second point would be Quan et al. 2002 study found that fibroblasts lose their regenerative properties as we age due to increasing Twist2. So Twist1 and Twist2 are very similar, but Twist1 is actually significantly longer by about 42 amino acids. So twist 1, 202 amino acids long, twist 2, 160. That smaller size of twist 2 makes twist 2 even harder to target because essentially there are less amino acids that we can try to bind to. So while it's likely that a solution will come from twist 1 first, we need to keep twist 2 in our minds as we're looking at this big picture of what causes hair loss.
After the twist brother-sister combo, we have PI1. PI1 is consistently stayed at the top in the online hair loss research community. A lot of our smartest researchers very excited about the target and currently testing to see how efficacious it really is. From prior work, both in the longevity and hair space, the current belief is that we want about 50 to 60% knockdown of the target. Although there are some counter arguments of people saying that maybe we don't want to touch the target, and we'll get into both real quick. On the case for downregulation, obviously Arion Therapeutics found very strong data in mice, or even better, human scalp skin on a mouse's back, and subsequently raised a lot of money to fund clinical trials of their ETO2 and ETO3 drugs, which are both PI1 inhibitors. There are also recent reports that minoxidil may normalize PI1, so bring it down from an upper yield level. And then even going back to some old studies in the late 90s, mutant mice that overexpress PI1 showed delayed antigen entry, which again shows that this drug may downregulate, it may promote antigen and inhibit catagen, which is obviously what we want. There are some researchers who are making the case against PI1. So Arion's patent has been published since 2020 and was obviously submitted a few years before that, presumably. And some people do believe that it is surprising that they could have such strong hair growth stimulation effects, but that their work still hasn't been peer reviewed, or published in a decent journal. So essentially, that these results may have been conducted in a shady lab, and that there may have been ulterior motives at play. And then on the PI, and then on the science side, PI1 is also very highly transcribed in rat antigen hair follicles. Again, rats, mice, very different from humans, but at the same time, still reason to maybe be wary. And then lastly, there are risks if we downregulate PI1. Again, similar to twist 1 in the wound healing department. If we downregulate PI1 too much, there may be a risk that we have excessive blood flow, which, if there's an injury, can lead to excessive bleeding. You can think of it kind of like as making your tissues looser. So if you're susceptible to arthritis or other things of that nature, it could also be a risk there. But at the same time, there was a leukemia study where patients with leukemia were taking a PI1 inhibitor for 12 months, and none saw very, very serious side effects. And the side effects that were seen were ended after the patients discontinued the drug.
Yeah, so next up we have YAP1. YAP1 is upregulated when there's wounding of the skin during the scarring process. So really important to understand, microscarring is largely correlated with loss of hair, so microscarring in the scalp. And then also where this is really important is actually in the case of hair transplants, where you're extracting hairs from the donor area, the back of the scalp. And typically what happens where you remove the hair, there's scarring formation that happens. So big shout out to Indana here, who's gotten Herdao in touch with Dr. Talib Barghouthi out of Jordan, who is actually testing verteporfin on his hair transplant patients. And the results do look encouraging from the pictures, where instead of scarring in the donor area, you're actually seeing follicle neogenesis and hair regrowth, which is really important because if you can regrow hairs in the donor area, you could theoretically get unlimited hair transplants to fill up as much of your bald spots on the top and front of your head as possible. To give a bit of the counterpoint to all of this positivity on verteporfin, I need to include Jumpman's point that verteporfin is photoactivated in order to actually inhibit YAP, so it may not be as effective as other molecules. And the second thing is that verteporfin might be too big of a molecule to actually get through the scalp and have the desired effects. So while there is positivity with verteporfin, we would encourage and we would actually plead, we want the community to keep looking for small molecules that actually inhibit YAP1 even more, might be even actually more effective, but serve sort of the same goal that people are trying with verteporfin.
Yeah, next up, TGF beta. While not a direct target due to its overarching effects and biphasic nature, um, which really means that some TGF beta is good for our hair follicles. If we bring TGF beta to zero, that would be bad. But upregulated TGF beta past a certain threshold, which I think we still need to define precisely what that threshold is, does further worsen this feedback loop that Jumpman's beautifully described, the Jumpman chain of events. There's a positive feedback loop between TGF beta and both TWIS1 and PI1. Obviously, two of our favorite targets from before. And really, that is a strong belief for why we're going after those targets. So even if TGF beta isn't a direct target that we're pursuing, we will be measuring it closely, especially as we're pursuing some of these other targets to see, are they generating the effect on TGF beta that we would suspect, as well as other proteins. But again, TGF beta really is at the top of the list there.
Okay, next up, FGF5. Of the fibroblast growth factors, FGF5 has the strongest association with androgenic alopecia. Knockout mice exhibited extended anagen, so knockout of FGF5 equaled longer growth phase of the hair follicle. We still haven't found a direct way to target FGF5 in vivo humans, which is just another reason to emphasize the siRNA and ASO antisense oligonucleotide screening that we're pursuing with some of the top labs in the world. So really exciting there.
Next up, we have BMP2, another target that has strong sides both for upregulation and downregulation. The downregulation group points to the fact that BMP inhibitors such as noggin and cordon are actually implicated in hair regrowth. And also BMP upregulation, not downregulation, but upregulation, may inhibit Wnt, which obviously we don't necessarily want. On the other side with BMP2 upregulation is that it downregulates PAX1. PAX1 didn't make our list yet, but we should maybe expect to see it soon. Downregulating PAX1 is a very desirable trait as deemed by Mr. Jumpman, and it's also is a key marker of TWIS1 downregulation. So because TWIS1 is very difficult to drug, BMP2 upregulation may actually be our best chance at generating a similar effect to downregulating TWIS1. So yeah, again, our BMP2 work is largely hindered on our TWIS1 work, which I think emphasizes our need to fund our TWIS1 work as quickly as possible.
Next up, we have ZNRF3 and RNF3. Both of those proteins are Wnt receptor degraders. So inhibition of both of those would dramatically upregulate the Wnt pathway, stimulating the growth of hair follicles. As we've touched on earlier throughout this target list, and as many hair loss researchers, new hair loss researchers quickly learn, they think, regrow hair is as simple as upregulating Wnt. Unfortunately, Wnt is the ultimate double-edged sword, where the more you upregulate, yes, the more hair growth you get, but you also significantly increase chances of causing cancer somewhere in the body, which makes this sort of treatment sort of a no-fly zone for many patients. As we'll touch on in a bit, this is just another case for perfectly localized delivery, which ultimately I think vastly expands the scope of treatments that we could actually start testing on patients.
Next up, we have OTS2. OTS2 downregulation is another mechanism that has shown very similar effects to TWIS1 downregulation. So again, just really further emphasizing the need for our TWIS1 work. Please don't make memes of me as a TWIS1 fanboy, but I am. The OTS2 target is largely unexplored outside of the brain, but as our work with thyroid hormones has told us, the brain and the hair follicle share many commonalities, and so it could be exciting to see if we can continue to find more brain proteins that impact our skin as well.
Next up, ALX4 inhibition upregulates sonic hedgehog signaling, which is a powerful overarching pathway you see throughout hair loss along with Wnt. And then it also upregulates NCAM expression. NCAM is a cellular adhesion molecule necessary for healthy dermal papilla functioning. We may likely get a lot of the similar positive effects of TWIS1 inhibition with ALX4 inhibition, as the two are correlated across several studies.
Yeah, and at the end of Jumpman's chain of events, downstream of all of these targets is mast cell degranulation, which, if you'll remember, Ralf was talking quite a bit about in our last Hairy Matters 3. Degranulation is when these immune cells called mast cells release histamine which you may have heard of if you have allergies, and matrix degrading enzymes. Basically, the matrix within your cells, these enzymes degrade that, the fibers and proteins that make that up, which cause our cells to become stiff or slowly cease to move. As our cells, hair cells, become more stiff, effectively you get into this, the negative fly loop starts all over, where you have this, the cell senses that something's wrong, it goes into panic mode, produces more reactive oxygen species, and the entire feedback loop continues. There are a lot of drugs that we know that target mast cells, and so while very little research has been done in using mast cell drugs in hair loss, and even though mast cells, we don't know which genes yet to target in mast cells, there is quite a big design space here for us to get into. So looking forward to getting into studies that involve mast cells as well. And we're live with Dr. Paus on TRH. First question, Dr. Paus, Potential Dingo, one of our prized community members from India, asks, what is the number one difference between T3 and T4?
Oh, we are talking about two different planets here. So thyroid hormones, T3, T4, they are the main metabolism hormones in your body. They basically crank up metabolism in all your cells. All your cells have receptors for them, for these hormones. And they are basically a ubiquitous hormone that every cell needs. And if you don't have enough of that, you die. TRH, different story. The thyroid gland is controlled by two hierarchies of controls. One sits in the pituitary gland, probably right here in the back, and the other one a little bit above in the brain, in an old part of the brain, the hypothalamus. And that part makes TRH. And it's an ancient tripeptide, 3 amino acids, neurohormone that controls the production of thyroid hormones, but talks to very different receptors, which are much less ubiquitously distributed. So much fewer cells and tissues respond to TRH directly. And it just so happens that we discovered that your scalp hair follicles are making not thyroid hormone made here by the thyroid gland, but the boss of the thyroid gland, TRH, and have receptors for it. And that's probably the most exciting part. This tripeptide neurohormone also stimulates hair growth more than any other neuromediator we have ever tested. And not only does it do that, it also selectively stimulates the pigmentation in your hair follicles. So if you're losing your hair and you're beginning to gray, happens in your 30s, don't forget that, until you get as far gone as me, that's a couple of decades along the road. But it begins usually in the 30s. So you're having a hormone that also directly stimulates exclusively the pigment cells in your hair follicles, the melanocytes of the hair follicle pigmentary unit, and it promotes hair growth, and it does that via very different mechanisms than thyroid hormones do. So the idea is, let's take this TRH and topically apply it, because you don't want to systemically apply it, because then you might overstimulate the thyroid gland. You don't want to do that. And we found out only recently, actually, that at least in some countries, TRH can be topically applied as a cosmetic substance, while thyroid hormones would always be a drug, because it's known to be such a potent hormone. It will never slip through as a cosmetic agent, while, to my own amazement, TRH might well be. It's actually on the list of permitted cosmetic ingredients, COSIC. And so that means we could perhaps even synergize here two completely different hormone systems that in your body are talking to each other all the time. And to close the loop, why TRH is so sexy, in addition, we discovered for the first time that while for thyroid hormones it had long been known that they stimulate your mitochondria, the energy metabolism factories in your cells, that TRH does that too, but probably again via very different mechanisms. So since hair production and maintaining a very actively growing, pigmented, large scalp hair follicle requires a huge amount of energy, that may be one of the reasons why the follicle makes TRH. It actually is so smart to give itself an injection in the arm all the time with this neurohormone to keep going, to keep pigmenting, and to remain fully energized. So we are getting the best from two different planets here that are, however, circling around each other already in real life, and that's why I'm advocating to now to put for the very first time ever in not only hair research but also in skin research to topically apply TRH to human scalp skin and see what happens. The problem is always, can it get through? Does it really make it deeply into the hair follicle? Luckily for thyroid hormones, we already know now from our first set of data we generated in our first HairDAO project that they do get down if you topically apply it. It may be more difficult with a tripeptide hormone, but if you use the right vehicle, and we have some ideas which ones to use, the hope is that we will get it down. And if we then the next step is, what if we combine the two, right? Thyroid hormones and TRH. Would we then even get not only an additive, perhaps even a synergistic effect? And then we will be in heaven.
Maybe just getting to the past research a little bit more. You've run, obviously before our human scalp skin study with T3, T4, both microdissected hair follicle studies for both T3, T4, and TRH. Do you mind just going a little bit more into the differences there that you saw between the different drugs?
Yes, there were differences and commonalities. So both promoted hair growth, they prolonged anagen, they stimulated the proliferation of hair matrix keratinocytes in the so-called anagen hair bulb, and they both stimulated hair pigmentation. But TRH did all of this more. Then if we looked, in those days we used, when we did these studies, microarray, not RNA sequencing, so we may have missed a lot of genes. But the genes that they induced were different ones. They were not the same. They shared some, and the ones they shared most were, interestingly, hair keratins and mitochondrial genes. But there were other genes that were very different. And that only goes to show or goes to support further the argument, even though it's very well known that the signal transduction mechanisms of both hormones are very, very different. Thyroid hormones work via a so-called nuclear receptor that sits in the cytoplasm and then moves into the nucleus, where it talks to genes that in their promoter region have a special response element that only this complex of thyroid hormone and nuclear hormone receptor can dock onto, while TRH works completely differently. It talks to a receptor on the cell surface, a transmembrane receptor, which then sends secondary signals and finally ends up sending a signal to the nucleus. Very, very different. And therefore, it was no surprise that there were many, many differentially expressed genes between the two groups of hormones. And we were actually more surprised how many commonalities there were. So we think these two groups of hormones are a perfect duo.
Yeah, terrific. Yeah, I mean, obviously we're very excited about the T3 and T4 results and future research into TRH. But if you could show one benefit of TRH over T3, T4, what would it be?
Yeah. For example, that's actually, I forgot to say that when you asked me the question before. There is a big difference in the half-life of thyroid hormones and TRH. TRH has a much shorter half-life than thyroid hormones. Therefore, the little bit that may get into the blood will be degraded much faster than thyroid hormones. So that's another plus that recommends topical application of TRH. For example, if we find, I'll give you just one example, if we find that in the study that I'm suggesting, that topically applied TRH is as effective in promoting hair growth in human scalp after topical application as thyroid hormones, and you would ask me, so what would you start a study with? Then I would say, start with TRH. In the meantime, we test on the scalp in the lab in organ culture, is the combination better? And then you check in a clinical trial how much actually makes it into the blood in this combination of each individual compound.
Ralf also sort of takes issue with the porcine ear model that we're using, which I understand. Look, if we're able to get a ton of human scalp skin in vitro to test on, that's my favorite model. I'd love to use that. But ultimately, the way I think our community is thinking about this is the pig's ear model will be reproducible over the largest amount of delivery vehicles. There's a huge potential for a very cheap cost to essentially test the delivery of anywhere up to 50 vehicles loaded with dutasteride on the pig's ear. Once we see what's most effective in the pig's ear, we can then go to the more expensive model, which is the human scalp skin model. If Ralf says that he has 100 samples of scalp skin for us to use, I'm all for jumping straight to the human scalp skin, but I'm not sure that's the case. So with that, we'll take it away. Ralf has obviously been doing this for much longer than any of us, and so respect whatever he has to say immensely.
Obviously, as Averbs was saying, there's been significant debate on the model we should use for our delivery vehicle study. In your own words, as quickly as possible, what do you think of the porcine ear model?
Oh, the porcine ear model. Yeah, so that porcine ear model. Of course, the porcine ear model is very, very much being pushed by everyone who works with that model. The scientists do that, right? They always think the models they work with themselves must be the best in the universe. This one is not for hair research, and I know what I'm talking about because actually the first company that I founded was based on a pig's ear skin model that we developed ourselves, and quite successfully so. So there are some things the pigs. Let me give you the background. Why people are even interested in pig skin? So the problem is that the cosmetic industry at one point in time was not allowed anymore to do any animal experimentation, right? Because that was considered unethical. Now it's the customer who's the guinea pig when you try a cosmetic compound, right? But what you can still use is slaughter material, because these animals will not be specifically killed for the purpose of testing something. They will be killed for the purpose of consumption. And then the leftovers, and pig ears are only popular in some countries, they can be used for whatever you want. And it's not considered an animal experiment, because the animal is long dead by the time you look at the ear. So then that's one thing. There was basically a political, economic reason for wanting to use slaughter material from animals. And pig skin has one thing really going for it, that it's generally thought to be closest to human skin, much closer to human skin than mouse skin. And that is true. It's not as close as non-human primate skins, also apes, stump-tailed macaque and all that. And you could never claim that stump-tailed macaque could be used as slaughter material for testing cosmetic agents unless you're in China, right? Where non-human primates are consumed. So there's another argument why porcine skin, pig skin has been advocated as being wonderful for testing cosmetic agents or topically applied drugs, and that is that the tension of the skin is higher than if you take a piece of human skin and you put it in culture, then it relaxes. But since the dermis of the pig skin is very thick, that basically extends or heightens the tension. That then makes it easier for the follicular canals, the most distal part of the hair follicle, where the hair follicle moves into the epidermis, to remain a bit wider open. That's true. So you get a kind of shrinkage effect with scalp skin, human scalp skin, that you put in organ culture. But it's the very same thick dermis of the pig skin that also gives you false readouts, because of course, one of the things you want to know, how much of what I topically administer to the scalp will actually end up in the blood. And if you have a very thick dermis, not only does that compound have to travel a larger distance, it also gets trapped in all the extracellular matrix proteins that make your dermis, all the collagens and multiple other matrix proteins that are located there. So you can get a very misleading readout how much actually makes it. into the blood. How do we do this in scalp skin? The scalp skin, you just administer it as you would do to pig skin in the top of the chunk of skin that you organ culture, and then you measure in the culture medium how much makes it into the culture medium. And while both models are not perfused, and that in itself brings an error element into the equation, you have a much higher likelihood of seeing in the human scalp skin model what ends up in the medium, and therefore as a predictive data point for how much might end up in a perfused scalp skin in a living human being. But there's something else, and to me that's the killer argument against using porcine skin if you don't have to, because you have nothing else. We don't have that problem because we have the real thing. If you want to know how a drug or a cosmetic agent or a nutraceutical works on human hair follicles living in their natural habitat on scalp, how can anything possibly be more predictive than the thing itself, right? So yes, it will be wonderful. Let's say you want to test topical dutasteride in porcine skin, and you show the porcine hair follicle responds beautifully. Does that give you any guarantee that the human hair follicle will do the same in the same vehicle? No, it doesn't. If you do the same with scalp skin, you still have no 100% guarantee that you will see the same thing on a living human being, but you're getting much closer than testing this in porcine skin. So in short, porcine skin is a wonderful thing to have if you have nothing else. If you do have the real thing, though, go for it.
Moving past that, there is a conception that if you have a small molecule that is above a certain size, 500 Daltons is deemed the threshold, is that it will sit inside the hair bulb and won't be systemically absorbed. Obviously, T3 and T4 both exceed that size, as does dutasteride. Is there any validity to this?
No, I think this is Andrew. This is a good point. So molecular size can give you a certain level of safety about what makes it into the system or not. But the problem is the larger the size, and we have seen that with all the nanoparticles and even nanocrystals one looks at, they just get stuck in the distal hair follicle. So they get stuck in the area of the follicle where it cannot have much impact on growth at all, because growth happens deep down in the lower follicle where it's embedded in your subcutaneous fat. And so size, if it does give you more safety, it also increases almost exponentially the likelihood that you will encounter huge difficulties of getting the stuff down there. So that's one problem. So that's basically, size is counterbalanced by the likelihood of getting the stuff to where you need to get it. And for androgenetic alopecia, you always need to get primarily, yes, you can, if you have a little impact on the bulge, the stem cell region, that will be good, which is fairly high up in the follicle, right? But primarily you need to hit the anagen hair bulb. And in a miniaturized hair follicle, that sits also closer to the skin surface than in a full-length hair follicle, and you want to target miniaturized hair follicles, but you also want to stop the full-length hair follicles, the terminal hair follicles, from becoming miniaturized. So you actually want to hit the scalp in the stage as it is in right now on your scalp, Andrew. When you still have a ton of terminal hair follicles, and you want to preserve those guys, right? But if you can't even get your test agent down into the upper subcutis, it's called the dermal white adipose tissue, then you have a problem. There's something else that's always underestimated. So size is okay, it's important, but it's also important how lipophilic your compound is. The more it loves water, the more difficult it is to get it into any tissue. For example, TRH is very hydrophilic, water loving. T3, T4 are pretty cool. They make it much easier on you to get them deep down into the tissue. And I don't know exactly what the what the characteristics for dutasteride are there in this case. But so it's the efficacy of your agent will be determined by a very complex equation into which many different factors have to be factored in. Size is only one of them. Hydrophilicity versus lipophilicity is another one. Charge is another one. Then another very important one is, does it get degraded? How much does it get degraded? And are all the enzymes that are responsible for degradation present in the skin or in your follicles? And the follicle is a little bit like a liver. It metabolizes an enormous a number of things, many drugs as well. It has all the enzymes for it. So in the end, if your compound beautifully penetrates, but then gets degraded, right? You have a problem. If then the degradation products, at least one of them, is still active, you don't have much of a problem. But if the degradation products are inactive, you have a problem. And for TRH, we are dealing with primarily the problem that it's pretty hydrophilic, so water loving, and there are not that many enzymes that degrade this specific tripeptide hormone. It's actually one of the, if not the most stable peptide hormone in the body. It has a kind of cyclic structure, so it's a bit protected from degradation, but we have to fight hard to get it down at the follicle. That's why this is a challenging project. Nobody has ever tried to get TRH into human skin, and certainly not into the follicle. But we think we know what kind of vehicle we have to test that gives us the highest likelihood of achieving exactly that. And then we can use it, hopefully, as a cosmetic agent, which will make it so much faster to bring it to the market.
Beautiful. And with the vehicle that you've ideated for TRH, are these vehicles customized? So no. This vehicle will only work for TRH? No. Or yeah, how? Excellent point.
So in a sense, they are customized for TRH, but they are not likely to work only for TRH. So once you have developed this vehicle, and we will base our experience with the on that we have acquired with other peptides. So we are not starting from scratch here. Otherwise, the project would take much longer. So what you do is you say which vehicles have already worked for other peptides that share some similarities, right? And then you just try that, and maybe you add something that enhances the penetration power, and maybe also makes it more difficult for degrading enzymes to attack your peptide of choice, in this case TRH. So we always work with vehicles where we already have prior experience that for this kind of compound, they have a higher likelihood to give us the desired effect. There's another aspect we always have to keep in mind when developing vehicles, and that is, are all the ingredients FDA licensed? Which makes it much easier if you want to administer a drug. To get fast approval, and if it's a cosmetic agent, are all the ingredients on the list of allowed cosmetic product ingredients. So that's another regulatory concern you always have to have, and of course we follow these concerns.
Very cool, very cool. Yeah, I wonder if we could try to reuse the vehicle for siRNA or ASOs or something also small.
Yeah.
Maybe not.
And we all have our limitations. I'm not a chemist, and there is an entire brotherhood or sisterhood of experts on vehicles, on topical application vehicles out there, and that's why I use one of the best of them as a consultant for Cotanion, and for any research questions we have, because that really is not my area of expertise. I've always been pretty bad in chemistry in school, so not my turf. Not my turf. There's a reason why I became a dermatologist, right? Hopeless as a chemist.
Yeah. Well, awesome, Ralf. I mean, that is there anything else you want to say?
Yeah. I mentioned dutasteride several times. So I heard there are many people in your community who are totally excited about squeezing dutasteride into pig skin, or as a first step to developing a more efficacious topically applicable form of this 5-alpha reductase inhibitor, which is stronger than finasteride. Yeah, when you started telling me about this, I started yawning. Now, this was like Valium. I mean, gee, you have come up with such an innovative organization, and then you come with these very old-fashioned, utterly boring, almost brain-dead projects. I mean, where is the excitement and innovation in that? No, so many companies have already tried that. So many research groups have already worked on that. Can we please be a bit more creative than that?
Definitely. Yeah.
I hope so.
Yeah. I mean, I think it's interesting. What's interesting to me, at least, is how if you can get any drug, right, not just dutasteride, but because this would completely change, because then cyclosporin could become a viable treatment if you could find a way to just have these drugs sit.
Yeah, or the one that I'm so excited about, tacrolimus FK506, right? That's actually the biggest problem there, is that the pharma companies that have developed these drugs had no interest, no serious interest in developing hair follicle targeting topical applications. So for tacrolimus, for example, they found an application that works on the epidermis. It makes it into the epidermis, and it's actually very effective for atopic dermatitis, and very safe, can be given to children, and so on, right? But for cyclosporin, it really has failed to make it to exert efficacy. It actually makes it into the skin somehow, but doesn't, for some reason, it doesn't really work there. So the challenge of developing truly hair follicle targeting vehicles that are at least enhancing the likelihood of enriching your active compound in the right regions of the follicle, so in the bulge, or most important in the bulb, is a challenge that we have not met yet. And any vehicle that has been suggested for that so far in the past, first there were hair follicle targeting liposomes, then there came nanosomes, then nanoparticles, nanocrystals. None of them has made it into a commercially viable product. Why is that? Well, probably because those preparations really either are too unstable in themselves. Liposomes are, for example, pretty unstable, or they just get stuck too high up. And nanoparticles probably do that. So our approach is a little bit different one. We are not taking nanoparticles or complicated liposomes. We actually take fairly simple ingredients that increase the lipophilicity of the agent that we want to deliver. And since everything that's fat loving, lipophilic, has a higher natural tendency to make it through the transfollicular penetration route. And so that's our general approach. And so far, our philosophy there has been keep these vehicles as simple as possible. That's good for their stability. That's good for production of the actual product, for a good shelf life of the product. That's also important, of course, and to keep the price low. That in the end is also a concern, right? You don't want to develop vehicles where the vehicle itself costs you $1,000, right? That would be a dead duck. We don't want that.
Unless that vehicle could hold tacrolimus, dutasteride, and somehow sit only in the bulb and not let the drug get into the bloodstream, right?
Yeah, but that is probably not going to happen. So we have to be realistic about something. So what if you manage to get any active agent deep down into the hair follicle, knowing how very well perfused the follicle is? It has a very dense network of blood vessels around it, and also a very interesting lymphatic vessel network that only now becomes slowly developed or discovered. The likelihood that if anything makes it so deep down into the follicle that at least a little bit, a tiny fraction, hopefully only, will make it into the bloodstream is high and not low. Then the question is, how fast does it then get degraded in the bloodstream? And when it first passes through the liver, will the liver immediately done the rest of the degradation? And then how much will then end up in organs where you don't want these substances to end up? But that you can really realistically only test in a clinical trial. And you do this by taking blood collections while you topically administer for a long time your test agent. And then you know how much ends up in the blood. And that is, in essence, the only reliable test you really have for getting a handle on that. But realism is a good thing here. We know that if we find super good vehicles, that also will increase the likelihood that a little bit ends up in the blood. That's inevitable. And even if you bind your test compound to a big particle that gets stuck in their follicle, well, it still has to get released from this big particle, right? And whatever is being released, a little fraction of that will also end up in the blood vessels around the hair follicle and thereby in the bloodstream. But if that is only 1% or 0.1%, you are normally below the levels of getting any pharmacologically relevant effects.
Yeah. Well, if only 1% or 0.1% of our topical dutasteride T3, T4, or any of the confidential projects that we're working on go systemic, then I think we like our chances. With that, kicking it back to the other Andrew, who's with Nicholas from Labdao.
Okay, we're here with Nicholas Rindorf, the co-founder of Labdao. Labdao is the organization that has also made the Plex platform, which we at Labdao are going to be using for drug design for all of the gene targets we've come up with. Thanks for coming on, Nicholas. Really excited to talk to you. Can you first just start off by giving us an overview of Labdao and Plex?
Awesome. Yeah. Well, thanks, Andrew, for having me. It's good to be here. Well, maybe I can start off just giving a broad overview about what Plex is, and then how it can help the Labdao community in particular at the current idea stage where you're at. So when we came together as a Labdao community, the whole idea was, let's create a place where we can trade scientific services online. And the idea was, that we should build an exchange where you can trade services ranging from computational time all the way to eventually laboratory services. And over the last months, we've really been focusing on computational services in particular. And what we've been building, and it's really been very closely coordinated with the Bacalao team at Protocol Labs that just recently actually spun out into Expanso, another company in this space, is compute over data and running complex compute jobs over a whole community network. So instead of having a university cluster that's very centralized, now everybody can sort of contribute some of their compute into a compute network that's basically owned by a large group of people. And Plex is the tool that allows you to tap into that community cluster and run compute jobs and also provide some of your compute. So that's what Plex is. All right, so let's take a step back and talk about drug discovery overall. So one idea that I would like to start off is I sometimes call it sort of the treatment effect almanac. So let's think about like a parallel universe where we have we know all the diseases. We have a list of them. They're ICD. We have all the diseases that we know, and then we have about on the one axis of this big Excel sheet that it's fully imaginary. We don't have it, but we have this like big Excel sheet where we have on the columns with all the diseases that we can think of, and in the row space we have all the small molecules that exist in the world, and there are a lot, right? There are like trillions of small potential chemicals that you can under the laws of physics from first principles generate. So there is this imaginary table, and you can call it an almanac, that exists. And the big problem is that generating an entry where you measure basically, given for this disease at this column, for this particular small molecule, what is the effect of this effect of this molecule for the given disease? measuring, like putting a number into that cell in the Excel sheet costs you hundreds of millions of dollars. And if you don't do it with additional, preliminary work, it's also highly unethical because people, might get hurt. So this is the problem that, all of drug discovery is around, is that this table, this Excel sheet exists in everybody's head, but filling in, like penciling in a number into one of these cells in the spreadsheet is extremely expensive. So we need to get smart about the way, in which we reduce the cost of filling in the number in that spreadsheet. And one column that the HairDAO community really cares about is hair loss, especially the most common form of hair loss that's related to androgen and androgen metabolism. Okay, so now how do we reduce the cost of filling out that that the cells in that column, where we could test every small molecule in the world, and everybody in the community would take it, but there would be a lot of cost that would be created and people would get hurt. So how can we be more efficient? And this is where we get into like the sort of preclinical drug discovery realm. And obviously you can give it to mice, or you can give them small molecules to cells. That's still pretty expensive. So is there something that you can do even before you go into cells? So before you even go into the physical lab? And this is what a lot of like computational drug discovery is trying to do. It's trying to make it easier to go through this, like all these different rows in this big table, and like trim it down so that we're only testing small molecules that we believe, it's worth spending our money on. And one way in which we can trim that down is if we know a target, like a particular gene or protein that's related with androgen-associated hair loss, and we try to inhibit that, for example. And usually the way that you would look for a drug that inhibits that gene or protein would be to do a high-throughput drug screen. So you actually are in the lab and you actually synthesize the molecule and you see whether it inhibits the protein's function. Now that itself is also really expensive, and there are approaches to try to actually do that type of simulation of whether a given small molecule docks tightly to a protein on your computer.
Okay, yeah. So Nicholas, as I would see it, there's sort of three main components to in silico drug design. First, you have target identification of the gene that you're looking to upregulate or downregulate. Then you have a decision to make on which software you want to use to predict the folding or the protein structure. Obviously, if there's an observable structure, you can just use that. And then finally, there's the decision to make which software to use to predict the docking of whatever drug you're trying to test against that protein. Now, the first part of this funnel, other companies like in silico medicine are using large language models to predict certain gene targets. It seems like you guys at Labdao are actually building with DeSci in mind and making the bet that community might actually be better sort of target identifiers than any sort of AI on the marketplace. Is that true?
Yeah, I mean, let's take a step back briefly and just talk about the key steps when we try to use computers to reduce the search space. So the first step is this target identification step. You need to know what you're looking for when you do computational drug discovery. So ideally, for example, a protein that's related with the disease process. And there are 20,000 proteins in the human body, so you need to reduce your search space also around these proteins. And that's what I believe communities are really good at. You can use large language models or different sort of target discovery suites that try to surface new targets, but most of the time, I believe the most promising targets have already been discussed in the literature, and groups of humans that care about a given disease that come together from all different regions of the world, from all different walks of life, and they scavenge the internet because they're interested in something. I think we shouldn't underestimate the sort of web crawler that is Homo sapiens and that is a social community. These are the ultimate sort of query tool community. So I think if one thing that I'm really optimistic about, then it's the power of humans to retrieve information and process it. So I think that's what we lean into at Labdao, is that we try to really empower scientific communities, because we do believe that the best ideas come out of communities that have very different viewpoints, and they bring their knowledge together. I think that's the first piece. Now I can go a bit more into the methods in just a second as well. So now, once you have identified the target, you go on and you say, well, let's look at the structure of that target. Most of the time, today actually there is some structural information available for the really very relevant targets, but in some cases there's no structure available for a protein, for example, that you're trying to inhibit using a small molecule. So you do need to make certain predictions around what the structure of a protein looks like. It's active research how good these types of structure predictions tools are, and frankly there's some evidence suggesting that if you predict the structure of the protein and then you try to do this docking where you have the lock, you folded the lock and you're looking for different keys to inhibit it, that can be that can be actually pretty challenging. So we're actively looking into different docking tools that sort of try to look for keys and see how well do they perform if we have structures that have already been measured in the lab and if we have structures that we completely predict.
Okay, so yeah, Nicholas. So then on the sort of the second part of this overall design process, Labdao seems to have made the decision to use CollabFold instead of AlphaFold, which I think a lot more people have actually heard about. What went into that decision? Why did you guys decide to do that?
So in principle, you can run any type of tool on Plex, but one reason why we've leaned into CollabFold was because it's way more lightweight to run. It has the same model. It's basically the same model. It is AlphaFold, but it does one important thing differently. So when you think about how AlphaFold works, it has two types of core processes. You give it a sequence for a protein that you want to construct. And what it does under the hood is it runs something that's called a multiple sequence alignment. So it goes basically into databases where we collect all the types of protein sequences or genetic sequences for that matter that humankind has collected. We do a huge search and we basically align the protein that you want to determine the structure of to all sequences that we've deposited as a society. And then we can use certain patterns in the way that amino acids, so basically the building blocks of proteins, vary to improve the performance of the algorithm. Okay, now here's the kicker. When you do this with AlphaFold, you need to have this database on your computer. And those are this at least 2 terabytes of data that you need to have on your computer to run a database query against. And there is a scientist out of Singapore, Martin Steinegger, that saw this, and he was that saw AlphaFold coming out, and he was also maintaining a website and an algorithm that's called MMSeq2. And MMSeq2 is a really, really fast website that allows you to query these databases. But you don't have to download the database yourself. You can just use the web servers by I think it's the University of Singapore to do the querying. And then he said, well, that's pretty awesome. So I can run AlphaFold on something that's just as lightweight as a Google Colab. And hence we have CollabFold because it's AlphaFold just, a bit faster in terms of the way that you query the multiple sequence alignment databases.
And as the final part of this whole design process, what's the favorite docking tool? What does Labdao typically push people towards in terms of predicting which molecules will bind to the proteins?
Yeah, I think it's hard to say right now what's the favorite software. We've been trying different tools, and I think Labdao's mission is, or key idea is that open tools accelerate progress. So the more tools we put together online, the more tools we make discover, the more tools we can test, and we can see which ones is the best. There is some evidence suggesting that trained docking systems like DiffDock are surprisingly good at retaining accuracy when you actually predict the protein structure. So some of these traditional mechanistic docking systems, they see quite a degradation, basically a cliff in performance. They drop down when you go from an observed structure to a predicted structure, while some of these learned docking systems, they seem to be a bit more robust to that. Now, at the end of the day, we need to look at the data. We need to look at what, for a given project, do we have observed structures or do we have to predict structures? And then based on whether we have observed structures or predicted structures, we can use the best docking tool for the job. in some cases, we do have observed structures, and then we tend to go with the observed structures.
Okay, and then my next question is, what are Labdao's plans for monetization of the Plex platform?
Yeah, I think in terms of eventually creating value for the whole community, what we plan to do is, right now we have this Plex compute network, and we are running nodes, and right now through the association, we're running a couple nodes. But what we really want is the whole community to if you have spare compute, you can run a node and you can actually contribute to the compute power of the network. And eventually, obviously, if you contribute compute power to the network, there is an ability to for you to have a piece of ownership or governance power over that community and over the network, and how certain jobs are prioritized. So that's the way in which we really plan as a community to create value. Another method that we're looking into, and I cannot commit on anything right now, is if you can create a tool, and you create a tool to the network, right? There are nodes, there's compute, but there are also the tools. If you create a tool, what we want to make sure is that that work is recognized as well, and there's a way for you to also earn from putting in the effort and creating an open source tool for I think the broader scientific community. So those are the two primary ways in which I think the community will create value. I think that's pretty undeniable.
Yeah, I think our community is actually really excited for Hair Minds when they can contribute their compute in exchange for the Hair token.
Very, very much looking forward to that. Thanks so much for having me on the show. Always down to talk a bit more about specifics on projects that are relevant to HairDAO. I do believe, again, online communities are the best hotspots, shelling points of new ideas, and so very, very keen to support your research and try to take it to the next level. Yeah, we're going to be starting. I think prototype nodes, testnet nodes, probably towards the end of Q3. So stay in the loop for that. And if you want to if you have some spare compute, we'd love to have some people that are keen to tinker and get their hands dirty and run some compute themselves. Yeah, if you have some excess compute, old ETH miners, we'd love to talk.
Yeah, and then what kind of hardware do people actually need in order to contribute compute to your system?
Yeah, so the current configuration is we're currently doing this mostly on cloud internally. But when we look at sort of hardware that's at the edge, ideally you need to have an NVIDIA GPU. There are certain popular GPUs that people used to do for ETH mining. I think a lot of them should still work. And then you need to run Ubuntu. You need to have a Docker runtime. We have all of that already packaged. So just get in touch. If you have an NVIDIA GPU, you're good. Just get in touch. Yeah.
All right, Nicholas, thanks so much for coming on. Really excited to continue our work with Labdao. I think it's going to be an awesome partnership. Thanks again.
That's Harry Matters 4, is it not?
That's it. We're all done. So hope you guys enjoyed that. We want to keep finding gene targets, exploring the current gene targets. We need to figure out what we want to do with Ralf's TRH study. To fund or not to fund?
That's just the question.
Yeah, and then I think a lot of cool projects coming ahead with Labdao on the horizon. Anything else you should share?
Some exciting updates on the way. Nothing public.
All right, exciting updates on the horizon. Stay tuned for the next episode. See you guys later.